Hi everyone, in this video we are going to understand an important concept from genetics, that is the law of independent assortment. So in a monohybrid cross we study about two laws. They are law of dominance and law of segregation. In a dihybrid cross we study about another concept that is called a law of independent assortment. In this video we will understand what is this law of independent assortment uh, through an analogy from tossing of a coin. Okay, let's imagine that we have two coins. Each coin has one allele for one character. For example, coin 1 has a allele for one character, and coin 2 has a allele for the other character. And imagine that these alleles are just they code a character, and these alleles are present on different coins. And when we flip these coins, okay, the result of the coin A doesn't affect the result of the coin 2. Similarly, the result of coin 2 also doesn't affect the result of the coin A. On one side of the coin A, suppose we have allele A, and the other side, opposite side, we have the allele small a. On the other coin, second coin, there we have on one side capital B allele, on the opposite side we have the capital B allele. Okay, when we toss the coin 1 and coin 2 together, so the output may be a capital A and capital B, or capital A or small b, or small a capital B, or small a small b. That means we will have four outputs when we toss coins with this type of alleles. Okay, that means when these alleles are present on different coins, we get four types of outputs, or four combinations. Okay, similarly when we apply this to this dihybrid cross. So in dihybrid cross, when we perform a test cross, so suppose when we perform a test cross for a plant which is heterozygous for the two characters, heterozygous for the two characters, with a recessive plant for the same characters, so which has a genotype recessive for the same character. So we get the gametes where the combination of these alleles, suppose when we look into this heterozygous plant, so there we get four types of gametes. So in recessive type, there we get only one type of gamete. Okay, so when we perform a cross between these two, so we get offsprings. When we observe their phenotype, so we get all these progeny in a 1:1:1 ratio. That means all the phenotypes so have equal percentage uh, in their representation in the offspring. Okay, uh, so from this we can understand that these the output that we get, four types of the combinations from these coins. So that tells that, okay, this is possible when one allele doesn't affect the that doesn't affect the outcome of the other allele. So this is called the independent assortment. So law of independent assortment, where where one allele doesn't affect the other allele during the segregation in the meiosis 2. Okay, so next one is condition where, just imagine, these alleles are not present on the different chromosomes. Suppose they are present on the same chromosome. So this type of 1:1:1:1 ratio of phenotypes are possible when these genes or these alleles are present on the different chromosomes. Like we have two coins over here. Suppose we have genes on the same chromosome. For example, the two characters. So the genes with that codes these two characters are present on the same chromosome, like the kind that we can see here. On one side we have capital A and capital B. On the opposite side, on the side 2, we have a small a and small b. Just imagine that we have a coin like that. Okay, suppose when we perform a test cross, so of the individuals where the genes are present on the same chromosome. So when genes are present on the same chromosome, and we say that no, these genes are linked genes. Okay, and when we perform a test cross for in the same condition, so with this type of the genotype, so we get offsprings. So these offsprings, there we don't get 1 is to 1 is to 1 is to 1 ratio. So when we get 1 is to 1 is to 1 is to 1 ratio in all these four phenotypes, two phenotypes are similar to their parents, and that other two phenotypes are a new phenotypes that we observe, new combination, with a new combination, the two phenotypes are observed. So when we have this kind of arrangement of genes on the same chromosome, like capital A, capital B on one side of the coin, and on the opposite side is a small a and small b. So when you flip a coin, you get two outputs, so like capital A, capital B, or small a and small b, but we never get a combination like capital B with the small capital A with the small b, or small a with the capital B. We never get such a type of combination. Okay, so the parental combination like capital A, capital B, and small a, small b. So when we perform such type of test cross for this type of genotypes, okay, so we get only the parental conditions. So the parental phenotypes like capital A, capital B, and small a, small b, we get only 1 is to 1, just two phenotypes, and these phenotypes are the parental phenotypes. And from this we can understand that okay, these genes are present on the same chromosome, and they move together during the during the segregation of the chromosomes. So these chromosomes, that means these genes, since they are present on the same chromosome, so they move together, and they are inherited in the offsprings together. So this is called gene linkage. So when genes are present on the different chromosomes, so there we don't see the linkage. When the genes are present on the same chromosome, so we observe the linkage. So this is the concept of law of independent assortment. Thank you for watching this video.